Gastrointestinal cancers are one of the leading causes of death worldwide, account for about 9% of total cancer mortality. But of all the cancers affecting the gastrointestinal tract, the most worrying are gastric and colorectal because they have alarming rates. The greatest incidence are in eastern regions, mainly because of food habits. Cigarette smoking, hot food, and alcohol use are major risk factors, as well as hereditary genetic mutations. According to 2018 data published by GlobalCan, incidence levels for gastric and colorectal cancer are 5.7% and 10.2% of the total number of new cancer cases respectively, and mortality 8.2% for gastric cancer and 9.2% for colorectal. Many people who develop this condition suffer mutations in CDH1 gene, which encodes the protein encoding which has various functions such as cell adhesion, tumor suppression, and epithelial cell mobility and proliferation. When this gene mutated, the protein production is reduced and may even stop synthesizing, make room for the tumor to infiltrate it until the process of metastasis. The objective of this study was to evaluate the level of CDH1 gene expression in fresh biological samples for patients with gastrointestinal cancer. The methodology used to study gene expression levels was RNA extraction from intestinal, gastric, rectal, and esophageal tissues using the ReliaPrep RNA tissue extraction kit from Promega. After extractions, the extracted RNAs were converted to cDNA using the Qiagen Quantitech reverse kit from Qiagen. And finally, the PCR procedure was performed with the GoldTech PCR master mix kit from Promega. In total, 22 samples were used, but only 17 were successful in the research, as 4 didn't show good quality and 1 sample of esophageal was not expressed. As a result, we have all samples expression. The first graph shows the distribution of the audacities of each sample used between the analyzed subgroups, and it's possible to observe that there is not a statistical difference in the expression profile between the tissue types. The second graph shows the linear regression between the intestinal and gastric subgroups, and according to the data, it's not possible to verify that there is a linearity between the subgroups because R squared is not close to number 1. This indicates that further research is needed to compare the results obtained in patients with pain and benefits, and to relate these findings to the clinical trials studied so that the role of the CDH1 gene in the development of gastrointestinal cancer can be better elucidated.